breaking the wall of RNA therapeutics. How modified RNA drugs are reaching previously inaccessible parts of the body. Anastasia Khvorova, UMass Chan Medical School.
On November the 9th, 1989, I was in Moscow, watching with admiration and hope.
It's a great pleasure to be here, and what I will tell you today is a story of beauty in science, as well as breaking multiple walls to create a path towards transformative therapeutics for Huntington disease, as well as any other neurodegenerative diseases, and eventually aging of the brain. So this molecule is gorgeous. And it represents the way our cell control transfer of information. So life is information. And inside the nucleus, the information is being stored in double-stranded nucleic acid called DNA. And what you can see, it's a consequence of the letters. Yeah, blue, red, yellow, magenta, which is A, T, G, C. And every single one of them, your genomes are different, but they all consist from these different letters, which is information. Information is stored, but then it's need to be transferred, and it's being copied in a single-stranded molecule called mRNA, and Catherine will talk about making drugs out of those long mRNAs. It gets in a cytoplasm, and then it's need to be translated into function. So it's translated in a proteins, which is performing vast majority of functions inside the cell. Whenever such a complex informational process is happening, there is, of course, a lot of mistakes can happen. And cells develop a normal, natural mechanism. You're spending 5% of energy doing that, I'll call RNA interference or RNase-induced silencing complex. In this case, it can be programmed with small pieces of this information, like Google, which finds the targets and eliminates it from being able to translate it into the function. So practically, life is a storage of information, information transfer, and information interpretation into function. And every single disease, so 99% of diseases, it's either too much protein, not enough protein, or a wrong protein being made. And RNA interference mechanism, by blocking information transfer, provides a potential for a universal drug. So the human genome is sequenced, is known. It took us a while. It took over, the final version is available in 2022. But because the sequence of the genome is known, it's created an opportunity to actually utilize this natural mechanism to silence any gene on demand. I'm representing here RNA Therapeutics Institute, UMass Chan, and this is a premier institution which is contributing to discovery, understanding fundamental mechanism, as well as translating it into the drug, which is evidenced by Victor Ambros and Craig Miller actually receiving Nobel Prize for discovery of this fundamental mechanism, and Phil Zymer is the one who demonstrated that it can work in human cells. And the story you need to remember is that, yes, it's potentially a universal drug. As with any technology, whether it's fusion, whether it's AI, whether it's such a fundamental class of drugs, it went through the normal hype curve. What the hype curve means that something cool is discovered, everybody jumps on, things are moved into clinic, like big pharma is pouring money, and then nothing works. And when nothing works, what's happening? Everybody exits. Yeah, it's a very, you can see it with antibodies, with almost any break-in-the-wall technology. And when everybody exits, chemists, they come. And as a good friend of mine, Mana Marharan, who invented most of the chemistry platform in this field, saying, stay, if you don't know what to do, stay calm and do more chemistry. And indeed, this is what happened. And over the decade, the molecules get evolved. The reason why they failed initially, because the half-life of the fully natural siRNA in the blood of the starving student is around 5 seconds. And it's a negatively charged stick which doesn't go anywhere. So with dressing it in a complex chemical modification, we got it to the point that it's actually become a drug. And right now there's 7 approved drugs and many dozen in the final stages of clinical development. And all of that is because of the fundamental complex chemical innovation. So what is today in clinic is compounds are fully chemically modified. They're not RNA anymore. There's not a single RNA base left. It's a complex pattern, and then its structure where it's conjugated to the gallneck, which goes to the liver, or lipophilic entity, which broadly distribute, or antibody, or more complex architecture, that is what driving the delivery. The reason why full chemical stabilization is so important is that when those drugs get inside the cells, they're entrapped in cellular compartments called lysosomes, which is very, very aggressive for degradation of proteins. And as you see over here, it's like red siRNA entrapped in a green endosome. And when it's stabilized, those endosomes serve as our big friends because they provided an intracellular depot of the drug, which is being slowly released over time. And this is why in approved therapeutic today, think about it, how long do you think the clinical efficacy of the single administration? First time when I saw the data, it's really breaking the wall because it's up to a year. Think about it. It's a potential to change a daily pill, compliance, doctor's visits, prescription, pharmacy administration, to semi-vaccination to prevent your high cholesterol, where inclisiran, it's an existing approved drug today, which probably everybody over 50 should be taking, and vaccination against Alzheimer, making sure that you have tons of muscle, etc. Of course, all approved drugs right now is in the liver. And I got drugged into the field of Huntington by a good friend of mine, Neil Aroning, and he said, okay, Anastasia, you know, you need to deliver those things to the brain. And at the time, it was not only impossible task, it's a completely crazy thing. And Huntington is a devastated disorder which is caused by expansion of the CAG repeat in the first exon of the gene, and it's causing all those devastating consequences named here. But in a late age, people are born normal, they go to graduate school, they become famous politicians, journalists, and then at 40 or 80 age, it's a severe cognitive neuropsychiatric disorder with a very quick decline. So to be able to develop a drug for Huntington, you need to deliver it to the brain. And the 3 very talented students and postdocs working in my labs, they accidentally discover that if instead of one molecule, you link them together, you make them larger, you get a profound enhancement in the retention in the brain. And we got what is called pink brains. It's a brains which is turning pink, which is indicative of the siRNA drug being inside the brain. We got it in a large brain with essentially delivery to every single neuron. Okay, getting drug in the brain is a good thing, but you need to eliminate this protein which is causing function, and we were able to show that the Huntington protein is gone. Exciting. Yeah, I gave a talk in the Huntington Patient Disease Forum, and we got a standing ovation because there is finally a hope for a treatment. We start testing this compound in animal model of disease, and nothing happens. And that was super disappointing. Before there is another breakthrough, falling wall, it's a discovery by many groups indicating that in Huntington disease, the RNA and DNA, this information transfer, it's actually contributing to disease as much as a protein. And later on, we and others have figured out that when you have those expanded repeats, so it's repeated words, it's when information is being transferred, it's kind of misfold itself in the wrong way. And because it's misfold itself in the wrong way, natural mechanism of maintaining DNA integrity try to correct it. But when it's trying to correct something, it actually creates a more issue. And the issue is expanding of this repeat from 40 to 50, and when it's reached 150, the neuron dies. And this is why it's taken such a long time for disease to manifest. We figure out that if you silence the key target in this pathway called MSH3, you actually can block this expansion. So two processes, somatic repeat expansion and mutant huntingtin, it's contributing to the disease progression. So what do you do? There is a very nice model, animal model of the Huntington disease progression, which essentially over the months, several months, you can see this armadillo shape appear, which indicates there is more and more expansion. So there is expansion on the right. It also forms the protein aggregates you can see in patients as well, and most informally, the information is disturbed, like this transcriptional signature is all over the place, correct? So it's recaptures the features of the Huntington disease. So what we did next, it did a large study, which took a lot of money and a lot of time, and treated block for a year in this animal model expression of the disease protein, block somatic repeat expansion, all the controls, and a combination of the above. And what you can see is that if you look at this manifestation of the disease, those protein aggregates, if you remove the protein itself, nothing really is happening. If you block this genetic expansion, it reduces profoundly. And if you do both, the effects are cumulatively and profound, indicating that we might be up to something. But finally, we look at this distortion of the transcriptional signature. And what you see in a gray is the genes which change between disease, between patients and normal people. So there is 161 changes. It's like the wall, yeah, the gray wall. And when you block somatic repeat expansion, like 30% of the wall is gone. And when you do both, you are breaking 50% of the wall, which is potentially disease modifying. And mice, at least, are doing very, very well. So what I wanted to emphasize that when we are talking about the major breakthrough in the science, it's not one thing at a time. For development of the path towards transformative therapeutic for Huntington disease, and you understand it's an informational drug. The same technology right now is being used for treatment of Alzheimer, Parkinson, ALS, many orphan indications, and third of my lab is working on the rejuvenating of the brain. Of course, I don't want to age. But it requires like breakthrough discovery of RNAi, discovery of chemical stabilization, brain delivery, sequencing, Huntington gene, driver, combinatorial silencing, and this is providing a path towards the Huntington cure. And the last thing I wanted to say is that it really takes thousands of scientists in many countries working together on the fundamental basic things to get towards this type of transformative things and funding basic science. is very important, if not in US, at least in Germany. And thank you for a great team.